or good evening, or good afternoon, or good morning, depending on where you're joining us from today. On behalf of the SSR, I'm pleased to welcome you to today's webinar on male contraceptive development targeting spermatogenesis. So I'm Darryl Russell from the Robinson Research Institute at the University of Adelaide in Australia. So it's good morning from me. So I'm the chair of the organising committee for the SSR's Conference on Contraceptive Discovery, and I'll be the moderator for today's webinar. So I'd also like to introduce and thank Dr. Elizabeth Bromfield, who from the University of Newcastle, who will be my co-moderator. And we have participants today from all areas of reproductive biology and all geographic regions as well, who are interested in overcoming the challenges and creating innovations that improve the efficacy and access to contraceptive technologies. I'm also particularly excited to welcome and thank our speakers for today. We have a really outstanding lineup of speakers to talk to us about their research on contraceptive discovery. So just before we begin, just to let you know that all participants on the call are on mute throughout the call. But you can submit your questions through the question and answer feature that you'll see at the bottom of your screen. Click that question and answer and you'll be able to type your questions. Please don't use the chat feature, use the Q&A feature for your questions. And those questions will be read at the end of each presentation by our co-chair, Elizabeth Bromfield. So finally, the webinar today is being recorded and it will be made available to members on the SSR website after the call. So with those preliminaries out the way, let me introduce and thank our keynote speaker. That's Patricia Kosnikou from the Institute of Experimental Medicine, Argentina. And she's going to talk to us about CRISP proteins as novel targets for male contraceptive development. Thanks very much, Patty.
Thank you, Darryl. Okay, 1 second. Okay, here we are. Well, thank you, Darryl, and also Janice Evans for inviting me to present this lecture. And also thanks to the SSR and the Bill and Melinda Gates Foundation for supporting the organization of this meeting, which, as you know, was originally planned to be held in Argentina, but due to the pandemic, it became a virtual meeting, although keeping a very nice Argentine representation in the 3 days of the program. So thanks for this recognition to the science of our country, and also for giving me the opportunity to share with you the results we have obtained in our lab during the past years, working on the potential use of CRISP proteins as targets for male contraceptive development. So let's see if I can share. Okay, so, okay, it's okay?
There you go, that's good.
Good, thank you. Well, as you may know, 50% of the couples in reproductive age have fertility problems, indicating the need to develop new methods for both diagnosis and treatment of infertility. But at the same time, there are 40% of unwanted pregnancy that increases, that 20% of which end in abortion and increases a lot in developing countries. So this indicates the need to develop new contraceptive methods that meet the different needs and preferences of people in different parts of the world and at different stages of their reproductive lives. Which are the current contraceptive options? For women, we have the hormonal methods, the very well-known pill, which represent the revolution in the 60s because it allowed to uncouple sex from reproduction. And here I would like to mention two important women, Margaret Sanger, who was the one that proposed the project of the contraception method to Gregory Pincus, who was the investigator that finally developed the pill, and Catherine McCormick, who got the financial support for this project. So my little tribute to these two important women in the field of contraception. So we also have IUDs, diaphragm, cervical cap, sponge, female condom, and natural methods. But what do we have for men? We have just the condom, because vasectomy has 50% reversion, and you never know in which percentage you will fall. So it cannot be considered a contraceptive method. Interestingly, 1/3 of men in USA use condom or vasectomy. There are 500,000 vasectomies per year in that country, and 60 million vasectomized men in the world, indicating that there is clearly a niche in this field. And this, for this reason, the development of methods for male fertility regulation had been a very important issue in different international fora from the 70s, to emphasize the responsibility and promote the participation of men in family planning, meaning where or not to have children, when, how many, and how spaced. So why to develop male contraceptive methods? Because it's a family, in general, family planning must be shared by the two members of the couple. It's not a women issue, it's a couple issue. Second, because of health reasons. Women use contraceptive for 35 years, from menarche to menopause. Because it is a male right, we have to stop thinking that male contraception is to help women, and to start to think that male contraception empowers men and the couple. And finally, because there is a niche, as I indicated before, accordingly to the high number of vasectomized men in the world. Now, if a method is developed, of course, this will require cultural changes, but it's interesting to know that the acceptability level is at least 50% among men and higher among women. Nevertheless, if we want men to use contraceptive methods, we need to give them more options. A contraceptive method must be effective, safe, reversible, acceptable, easy to use, appropriate for different ages, and affordable. The development of male methods involve mainly hormonal and non-hormonal approaches. The hormonal methods have several advantages, like they use known and safe compounds, they are effective, and they are relatively non-expensive. But they have disadvantages too. The long time for reaching the azoospermia and for its recovery, around 3 months each, the permanent need of semen analysis to check the efficacy of the method, and the several side effects that the method can bring. And this is not a minor point, because as you know, side effects are less tolerated by men than by women, at least in the contraceptive field. The development of non-hormonal methods, on the other hand, is at a more basic research level and involve interference at 3 levels. Production in the testis, maturation in the epididymis, and the release of the sperm during ejaculation. And while the interference during production is really related to finding different ways to stop spermatogenesis, while the interference with the release during ejaculation mostly involve mechanical occlusion of the vas deferens, the interference with maturation is based on the fact that the sperm that leave the testes are immotile and unable to fertilize the egg, requiring to undergo a maturation process while they are passing through the epididymis to acquire progressive motility and the ability to recognize the egg. This is the fertilizing ability, mainly as a consequence of the association of epididymal proteins to the sperm plasma membrane. So the epididymis can be considered an excellent contraceptive target because there will no involve interference with the sperm or hormone production, which is so important for libido, but a specific interference with the acquisition of a sperm fertilizing ability. And the idea of having a ejaculate without the sperm fertilizing ability is a very safe, attractive, and promising approach. The reason why epididymal proteins become very good targets for fertility regulation. So the aims of our lab have been to study the role of epididymal proteins in sperm maturation and fertilization, and to explore their potential use for fertility regulation. So as you know, the fertilization process involves a first stage in which the sperm needs to pass through the mass of granulosa cells that surround the egg, known as cumulus ovorus. Then they sperm reach and bind to a glycoprotein matrix called the zona pellucida. Then it penetrates the zona, reaching what is called the perivitelline space, and finally fuses with the plasma membrane of the egg, also known as oolemma. And evidence indicates that these two stages, cumulus penetration and zona pellucida penetration, are mostly mediated by a strength provided by the hyperactivated motility, which is a very vigorous motility that the sperm develop in the oviduct and which is essential for the penetration of the egg coats. These two other steps of fertilization, zona pellucida binding and gamete fusion, are mainly mediated by the recognition mechanisms between complementary molecules in a ligand receptor manner. So the results I will show you today represent the contribution of our lab to try to elucidate the molecular mechanisms involved in these different stages of fertilization using a group of proteins known as CRISP, for cysteine-rich secretory proteins, characterized by the presence of 16 conserved cysteines, 10 of which are located in the C-terminal region of the molecule, called CRD for cysteine-rich domain, which has ion channel regulatory ability. And the 6 remaining cysteines are in the N-terminal domain, also known as PR1. This is a widely distributed and evolutionarily conserved family, as judged by the fact that members in mammals, in reptiles, more specifically in the venoms of the reptiles, and also in amphibians. And they're very homologous proteins in phylogenetically more distant individuals, such as insects, plants, and fungi, indicating that if these proteins remain through evolution, is because they must play important functional roles. In mammals, 4 proteins have been described, mainly in the male reproductive tract, CRISP1 synthesized in the epididymis, CRISP2 of testicular origin, CRISP3 with a wider distribution that involves reproductive and non-reproductive organs, and CRISP4 also synthesized in the epididymis. So what we know about the first member of the family? Well, it was identified in the rat epididymis and called DE by the lab of Waltheria in Argentina in 1976, where I did my PhD. It's a glycoprotein of 32 kD, synthesized in response to androgen. It associates with the sperm surface, both head and tail, during epididymal maturation, as we can see in this image. Now, evidence for our lab indicates that CRISP1 associates to the sperm surface with 2 different affinities, giving rise to 2 populations of CRISP1 in the gamete, one loosely and one strongly bound to the sperm surface, shown here in different colors. Now, this is what occurs in the male during maturation, but in the female, during capacitation, the loosely bound population is released from the sperm while acting as a decapacitating factor, while the strongly bound population remains on the sperm surface and participates in gamete interaction. More specifically, in these 2 stages of fertilization, zona pellucida binding and gamete fusion, through complementary sites localized in the zona pellucida and the oolemma, as we can see in this image. Now, structure function studies reveal, using the recombinant protein and also recombinant fragments and synthetic peptides, reveal that the ability of CRISP1 to bind to the oolemma resides in a very small region of only 12 amino acids within the PR1 domain, and which corresponds to one of the 2 signatures of the whole family, signature 2. Of note, there is a human CRISP1, which has 40% homology with rodent CRISP1. It's an epididymal glycoprotein of 30 kD. It's also associated with the sperm surface during epididymal maturation. And according to results from our lab, it also has a role in binding to the zona and gamete fusion through complementary sites in the human egg, as we can see here in the zona and in the oolemma. So having established the participation of CRISP1 in fertilization, we wonder whether the protein is relevant for fertility and therefore a good candidate for contraception. And we studied this by immunization of rats with CRIS1 and subsequent evaluation of their fertility. Why? Because immunization of animals is an easy and efficient method to neutralize a protein and analyze its relevance for fertility, allowing the identification of an immunocontraceptive target for the developing of a vaccine, or the identification of a contraceptive target in general to be blocked pharmacologically. So immunization of male and female rats with CRIS1 produced an immune response in both males and females, which was specific as judged by the fact that the antibodies recognized CRIS1 only in sperm and the epididymis and not in any of the other tissues and organs tested, and produced a significant inhibition of fertility in both males and females with no side effects at testicular or epididymal level. Similar results were obtained a few years later using the recombinant protein instead of the native protein for both male and female. And based on this, at present, we are studying the potential use of CRIS proteins for the development of a contraceptive vaccine for dogs with the collaboration and support of a national agency. So it seems like CRIS1 fulfills many of the requisites for a contraceptive target. It's an epididymal specific protein not involved in sperm hormone or sperm production. It is localized on the sperm surface and therefore accessible to immunological or drug attack. It has a role in fertilization. It is relevant for male and female fertility. It can be expressed as a functional recombinant protein, and it has a human epididymal homologue with the same localization and functional roles. So we then explore the relevance of this protein for fertility and for contraception by generating the knockout for CRIS1 in collaboration with Dr. Micheli from the NIH, and this was the first knockout for a CRIS family member. And histological examination revealed that the testes and the epididymis were normal, normal sperm number and normal sperm morphology, normal sperm motility, both fresh and capacitating, and normal acrosome reaction. However, when we analyzed the fertilizing ability of this sperm in vitro using SONA-free or SONA-intact eggs, we found a significant decrease in fertilization, consistent with the previously proposed roles of this protein in gamete fusion and SONA-Pelusi interaction, which in this case can be explained by the lack of the ligand to bind to the complementary sites. Now, in spite of these significant defects in the sperm fertilizing ability, the fertility of the knockout males was normal, and even when they were mated with knockout females, indicating that there are clear differences in fertility outcome by blocking CRIS1 in the adult animal by immunization or by deleting the CRIS1 gene in the knockout animal. And in this last case, suggesting that other proteins could be compensating the lack of CRIS1. So the best candidate was CRIS4, the other epididymal protein of the family. So we studied the relevance of CRIS4 for fertility and fertilization by generating the CRIS4 knockout mice. And we did this with the collaboration of Dr. Ikawa Onokabe from Osaka University in Japan. And again, the fertility of these animals was normal, but the fertilizing ability of the sperm in vitro using different types of sex was really very severely affected, as we can see here. And the results reveal that CRIS4 participates in both gamete fusion and SONA-Pelusi interaction. So we have epididymal CRIS1 that participates in SONA-Pelusi interaction and gamete fusion through ligand receptor interactions, and epididymal CRIS4 that participates in the same stages of fertilization, indicating that CRISP are multifunctional molecules involved in different stages of fertilization, and supporting the idea of a functional cooperation between homologous proteins in different stages of fertilization to ensure the success of fertilization. So at that point, we wonder what would happen if we generate the double CRIS1, CRIS4 knockout, with the idea that deleting more than one CRISP could lead to a stronger phenotype at fertility level. So at this point, I would like to mention that while in rodents, we have 4 CRISPR proteins, 2 of them of epidemial origin. In human, we have only 3 proteins and only 1 is expressed in the epididymis. And as I mentioned to you before, this protein participates in the same stages of fertilization that the rodent protein. The reason why human CRISP1 is considered the equivalent to the combination of the 2 rodent counterparts. And in this way, the double knockout CRISP1 and CRISP4 would give us an idea of what would happen in a man lacking or with mutations in the human CRISP1 protein. So we generate the double knockout by mating the individual CRISP1 and CRISP4 colonies, both fertile, and study the fertility and in vivo fertilization in these animals. And what we found is that for the first time, we found a significant inhibition in fertility, which correlate very well with the percentage of fertilized eggs in the ampulla, indicating that this should be the main responsible of the decrease in fertility in these animals. So these results, on one hand, confirm the relevance of CRISP protein for fertility and also the existence of compensatory mechanisms between CRISP1 and CRISP4. And at that point, we wonder what would happen if we delete more than 2 CRISP proteins. And so we decided to develop multiple knockouts for CRISP proteins by the CRISPR-Cas9 technology that, as you may know, today won the Nobel Prize in Chemistry. So the strategy we used to generate the multiple CRISP knockout mice was to design 2 guiding RNAs that target exon 2 in CRISP1 and CRISP3 genes, which have a high sequence identity and are located very close in the same chromosome. So these guiding RNAs were microinjected in zygotes from the CRISP2, CRISP4 double knockout colony that we generate by mating the CRISP2 and CRISP4 individual colonies that are available at our lab. And since CRISP1, 2, and 3 are in the same chromosome and CRISP4 in another one, these allow the generation of both the triple knockout lacking CRISP1, 2, and 3, and the quadruple knockout lacking the 4 proteins of the family. So we studied the fertility of these animals by mating the multiple knockouts with control females, observing a significant decrease in fertility in both groups, indicating that mice lacking 3 CRISP, 1, 2, and 3 were already infertile. And these have potential clinical implications because human CRISP1, 2, and 3 genes are clustered in a region within the same chromosome, so their expression could well be simultaneously affected. Now, as the percentage of fertilization in the ampulla was severely inhibited in both groups, we conclude that in vivo fertilization defects were the main responsible for the infertility of these triple and quadruple knockouts. Now, this in vivo fertilization failure could be due to sperm defects to reach the egg, that means transport problems, or to sperm defects to fertilize the egg, that means gamete interaction defects. So to study the transport defects, what we did was to mate the multiple knockouts with control females, and 4 hours later, we removed the sperm from the uterus in the uterine fluid with no dilution, and we observed them under the microscope. And we observed that while control of them were moving freely in the uterine fluid, the multiple knockouts were mostly immotile and trapped in aggregates. So we can conclude that triple and quadruple sperm exhibit clear motility defects within the uterus. Now, as another approach to study sperm transport defects, we study sperm migration within the oviduct. And we did this again by mating the males with control females and studying the presence of sperm in the different regions of the oviduct. That is, the lower isthmus close to the uterus, the middle isthmus, and the upper isthmus close to the ampulla. And we could do this taking advantage of the presence of the green fluorescent protein in these colonies and the consequent presence of, the labeling in the acrosomes of the sperm. And what we found is that while in controls, the sperm were located in the lower isthmus, as we can see here, and almost reaching the middle isthmus. For the multiple knockout animals, there was almost no sperm in the lower isthmus, as we can see in this image at higher magnification. So these results indicate that the multiple knockout sperm exhibit clear defects to migrate within the oviduct. To study the sperm gamete interaction defects, we analyzed the fertilizing ability of the mutant animals by IVF using the 3 types of eggs. And we found severely affected fertilizing ability for both. So we can conclude that in vivo fertilization failure in the multiple knockout animals was due to a combination of both transport and gamete interaction defects. So as a summary of these results, we can say that while in the control fertile animals, the sperm were motile in the uterus, they could migrate and they could penetrate the eggs in the ampulla. For the multiple infertile animals, the sperm were mostly immotile in the uterus and trapped in aggregates. They have defects to migrate within the oviduct, and even if they reach the ampulla, they have a severely impaired fertilizing ability, consistent with the reported roles of CRISP in fertilization. So we can say that CRISP are involved in functional events besides gamete interaction that could also be good targets for contraception. So going back to the reported roles of CRISP in gamete interaction, we know that epididymal CRISP1 participates in gamete fusion through the signature 2 region in the PR1 domain. And we also know that epididymal CRISP4 has a key role in gamete fusion. So based on this, we initiate a project on the identification of compounds that could interfere with the roles of CRISP1 and CRISP4 in gamete fusion. We did this with the support of MCI, Male Contraceptive Initiative, an organization that is contributing significantly to the field of male contraception, especially to the development of non-hormonal methods. So what we are doing is to develop a high throughput screening assay called Alpha Screen that senses the interaction between 2 molecules. It's a proximity assay. So in our case, the recombinant CRISP1 or CRISP4 proteins with a signature 2, and an antibody against the signature 2, anti-signature 2. So these 2 proteins are coupled to a donor and acceptor beads, and the system senses the interaction between the proteins by the transfer of a signal from the donor to the acceptor that is later detected in a plate wheel. So what we are doing is to use this assay to screen a compound library with the idea that those compounds that bind to the signature 2 will interfere with the interaction and therefore with the signal, allowing the identification of potential candidates to block gamete fusion and that's fertilization and fertility. And we are doing this with a collaboration of Ricardo Biondi in Argentina, who is a person with a great experience in the use of alpha screen, and who is helping a lot to achieve this goal. Now, in addition to the role of CRIS1 in gamete fusion through the signature 2, we found that the decapacitating role that I mentioned to you at the beginning of the talk was associated to the ion channel regulatory ability of these proteins mediated by the CRD domain. More specifically, what we found is that CRIS1 is a novel Caspr regulator that modulates sperm motility. As you know, Caspr is the main mammalian sperm calcium channel essential for male fertility in both rodents and humans. It is located in the principal piece of the tail. It is composed of several subunits, and it's involved in the development of hyperactivation, the very various motility that I mentioned to you before. And in the Caspr knockout sperm, there is no development of infertility, and this is the reason they are infertile. So, knowing that CRIS proteins have calcium channel regulatory ability, we studied the effect of CRIS1 on Caspr activity by patch clamp. And we did this with Alberto Darson from the Autonomous University of Mexico, who is an expert in electrophysiology in sperm. And we found a 50% inhibition in the Caspr currents when the sperm were exposed to the native protein, and no inhibition with the denatured CRIS1 protein, indicating that CRIS1 has the ability to inhibit Caspr, becoming the first natural inhibitor of the channel. And consistent with this, we found that exposure of a sperm to CRIS1 produced a significant decrease in several sperm parameters associated with hyperactivation, here, as well as in the total percentage of hyperactivation, indicating that CRIS1 inhibits both Caspr activity and hyperactivation development. So this result led us to propose that in the male, the loosely bound population binds to Caspr and inhibits its activity to avoid the expression of a premature hyperactivation in the male. But then in the female, CRIS1 is released during capacitation, allowing the activation of the channel and the development of hyperactivation motility that is essential for fertilization. So at present, we are studying the interaction between CRIS1 and Caspr with the idea of finding, identifying a compound that could mimic the inhibitory effect of CRIS1, binding to the channel, but with a higher affinity, strongly bound, leading to an irreversible inhibition of the channel and therefore to a potential male contraceptive method. So based on this, we initiate the collaboration with Dr. Xinshu Chung from Yale University, who, as you know, has made a significant contributions in the Caspr field and is helping us to try different strategies to go in this direction. Now, if we find a compound with this characteristic, the compound could also bind to Caspr in the female tract, leading to a female contraceptive method, indicating that this approach is like a unisex contraceptive method. So we can conclude that CRIS proteins are relevant for male fertility and novel targets for male contraceptive development. And we believe these studies will contribute not only to the identification of new targets for contraception, but also to a better understanding of the molecular mechanisms involved in mammalian fertilization, as well as to the development of new methods for both diagnosis and treatment of human infertility. I would like to finish by thanking the members of my group that have made a significant contribution to all the results that I show you today. Of course, to the national and international collaborators, and to the national and international agencies and organizations that have supported our work during these years. And also, of course, thank you for your attention. So thanks.
Fantastic talk, Patty. Thank you very much. What an impressive body of work. So we have many questions in the Q&A. I'll just remind everyone to please place your questions in the Q&A and not in the chat. Shall we start at the top? So we have a question from Janice. Do you think that the CRISP-1 immunization achieves reduced fertility by blocking CRISP-4 and not actually blocking CRISP-1, given that CRISP-1 is dispensable based on the knockout data? Is there evidence to support this or refute this?
Yeah, you mean in the, in the immunocontraceptive approach?
Yes, I believe that's what Janice is referring to, yes.
Yeah, okay. So it's possible because CRISP-1 and CRISP-4 are both in the surface and the possibility exists, but what we know is at least that what we found is that the antibodies do not cross-react with CRISP-4. The immune sera that we remove from the immunized animals do not cross-react with CRISP-4. So although it is a possibility in the sense that both are so similar, are in the surface, at least our results with the antibodies indicate that there's no cross-reaction. We don't know if we identify a compound, if the compound could also target CRISP-4, but the immune response is so specific that it is really due to CRISP-1.
Fantastic. Okay, so next we have another question. The in vitro assay indicated that single knockout sperm was impaired, yet the in vivo fertility is not impacted. What does this imply regarding the use of in vitro fertilization to assess sperm function? Can you see that one, Patty?
No, the problem is that I cannot, sorry, my Zoom has stopped, I don't know, so I cannot, sorry, 1 minute, we will see what happened here. Yeah, okay, so I think I'm back. Can you see my screen?
Yes.
Okay. So the question is, the in vitro assay indicate that the single knockout sperm was impaired, yet the in vivo fertility is not impaired. What does this imply regarding the use of in vitro fertilization to assess them? Well, really, what we found is that, you know, in vitro fertilization allows to see a lot of defects that you cannot see in vivo. And this is because the female tract is a very good selector of the best sperm. It is called the best semen analysis laboratory because it can really discriminate between the good ones and the excellent ones. So in vitro, you don't have that selection, and you have both the bad and the good sperm together competing for the egg. So in all these situations, you can see a lot of defects in vitro that you cannot see in vivo. But this is because of the different systems. The in vivo in some way can mask the problems that are expressed in vitro.
Very good. We have a question from Carmen. So the triple knockout and quadruple knockout phenotypes regarding sperm aggregation and decreased motility in the uterus and the failure to enter the oviduct, it highly resembles the uterine epithelial ER alpha knockout. The ER alpha knockout defect seems to be caused by alterations in protease secretion that lead to impaired semen liquefaction in the uterus. So do you think that CRISP proteins could contribute somehow to the effects of proteases in the uterus or have an alternative role in liquefaction?
Yeah, well, good question, Carmen. Well, we don't know, but what we found is that when you remove the sperm from the uterus, they were not only immotile and trapped in aggregates, but the fluid itself was more viscous. Possibility is, you know, that it is known that CRISP3 has roles. In general, CRISP1, 2, and 3. This is a recent report by the group of Gadella, have roles in oligomerization of the proteins and the formation of amyloid structures. So we believe that one possibility is that these proteins that are absent produce a deregulation of these processes of oligomerization or even liquefaction or coagulation that normally occurs. So it is possible that, this is due to the properties of the CRISP proteins. So we are exploring this, and in fact, we want to do this with the group of Bart Gadella. We have talked about that.
Very good. So a question from Dan Bernard. So given there are defects in both sperm transport, oh, where did that one go? There we go. Sperm transport and egg interactions, are you surprised that you don't see complete penetrance of the fertility defect in your quadruple knockout mice? Does this make you worried about targeting CRISP proteins, that they may not provide sufficient contraceptive efficacy?
Well, it is true that we don't have zero. No, we have a very, very low percentage of fertility with 50% sterile male, but we don't have zero. But as you saw, we believe that the pharmacological inhibition will be better, because when we did the immunization with only CRISP1, we got in vitro, we got significant inhibition, but the knockout, because of the plasticity of the system and the redundancy of the proteins, allow certain compensations that we didn't see in the immunization procedure, which is similar to the pharmacological procedure. So we believe that by using a pharmacological approach, we will probably have better results than with the knockout because of the plasticity of the system.
Very good, thank you. So question about CAT-SPR. So is the S2 sequence of CRISP1 involved in its interaction with CAT-SPR?
No, the signature 2 region is in the PR1 domain, and the ion channel activity is in the CRD domain.
Excellent. Next, could CRISP4 interfere with hyperactivation of sperm?
Yeah, CRISP4 we saw that in the knockout there is inhibition of hyperactivation with CRISP4 also. Yeah.
Fantastic. I might throw in a question if that's okay. So I was interested in whether there's an epithelial cell issue in the double knockout of CRISP1 and CRISP4, and whether you think that might affect immune privilege in the epididymis, especially in regards to using CRISPs as contraceptive targets. What's the outcome of that for immune privilege?
You mean the epithelial defects that we found, that we report?
Yeah, will there be an issue with the barrier? Yeah.
Yeah, well, this is the reason I mentioned that it is true that when you do a knockout, you have a lot of phenotypes that you don't have when you use a drug. We don't see anything when we apply, for example, the antibodies at the epididymal level, because these defects that at the epididymal epithelium level occur only in the knockout models. So although I cannot exclude the possibility that the use of both CRISP1 and CRISP4 proteins or drugs against these proteins could produce an effect in the epithelial, I think it is much more related to what you observe when you use the knockout technology.
Okay, fantastic. One very quick last question from Barcadella. Yes. So what is the phenotype of the single knockout for CRISP3?
Oh, good question. Unfortunately, we couldn't develop so far the single CRISP3 knockout. When we did it, we tried, of course, because it's the only single knockout of the family that is not available so far. But when we did it, we got a double knockout, 1-3. And what we found is that protein 3, CRISP3, is important for the expression of CRISP1. So we are now going to study why CRISP3 is important for the synthesis of CRISP1, but we need to get another approach in order to target just CRISP3 and not both proteins together. But the double knockout 1-3 has a very interesting phenotype also, and well, it is for reasons of time I couldn't show you the phenotype of the 1-3 double knockout, but it is in the paper that it's impressed in FACEP, so you can see it there.
Fantastic. Thank you very much, Patty, for a beautiful presentation.
Thank you.
I'll hand over to you, Daryl.
Yes, I stopped to share the screen.
Okay, thank you very much, Patty, for a fantastic presentation. And thanks to Patty and Liz for keeping us perfectly to time and thanks to everybody for a lot of interesting questions. There are questions that we didn't get to, but I know that Patty will be keen to try to get around to those people offline and give you answers to your questions. So you should see those forthcoming. Okay, so our next speaker is Professor Gustavo Blanco from the Kansas University Medical Center. And Gustavo is going to speak about targeting sodium-potassium ATPase alpha-4 for male contraception. Thank you very much, Gustavo.
Okay, thank you so much, Daryl. I would like to start by thanking Dr. Daryl Russell for inviting me for this. Also the organizing committee for giving me the opportunity to show our data here, and also the SSR for this great initiative. This is fantastic. So before I start, I will start with the acknowledgement. This is a project that has been ongoing for some time between my lab at University of Kansas Medical Center, and I put the names here of the two persons that have done most of the work, Jeff McDermott and Gladys Sanchez, and also our collaborator and a clinician, AJ Nangia. But this is a big collaboration with University of Minnesota and the laboratory of Gunda Georg, that is our medicinal chemist expert for the project. Also the support of NIH and NICHD and the contraceptive division. So we work on ion transport in sperm. And as you may know, sperm ions modify the function of sperm, and depending on the ion type and concentration where the sperm swims, you know, it can regulate function. And all these is done through a series of ion transporters at the plasma membrane of sperm. Some of them specifically regulate calcium, other ones pH, and other ones membrane potential. We specifically work on this transporter, the sodium-potassium ATPase. This protein is composed of two main subunits, an alpha that is shown here in gray, and a beta in red and yellow. And what this protein does is it uses the energy from the hydrolysis of ATP to internalize potassium and pump sodium out of the cell. It turns out that there are different isoforms of the sodium-potassium ATPase, and they are expressed in a tissue-specific manner. But the one that has the most limited pattern of expression is what is called the alpha-4 isoform, which is only expressed in testes and in sperm. It's not present in any other tissue. Here you can see an immunocytochemistry of seminiferous tubules in the testes, expressing alpha-4 in the adluminal side of the testes where the sperm is located. And then there is another isoform expressed from the 4 existing ones, alpha-1, 2, 3, and 4, but it's the alpha-1, which is the somatic form, and it's in all the seminiferous tubules. Here is a Western blot of the sperm, and the sperm contains the ubiquitous alpha-1 as well as alpha-4, but not alpha-2 or 3. Alpha-1 and alpha-4 have different localizations. Both are in the sperm flagellum, but alpha-4 mainly localized to the midpiece. Importantly, also alpha-4 is expressed late in spermatogenesis, and it's mainly found first in spermatids, and it's abundant in spermatozoa. We have detected these through antibodies or making a knocking mouse for the expression of GFP, which you can see here at the bottom, only can be seen in the adult testes or post-pubertal, and it has the same localization as that the antibody showed. So to study the function, we made a knockout mouse, and you have here the lack of expression of alpha-4 in the null mice. These mice are perfectly normal. They develop testes, they produce a sperm, but the males, and only the males, are completely infertile. Also, if you take sperm from these mice, the homozygous mice cannot fertilize eggs in vitro. And the main problem with this sperm is that compared to the wild type that swims normally, sperm from a knockout mouse barely moves, and the ones that move do it with a severe reduction in motility. Not only total motility, but all parameters of sperm motility are decreased, and specifically hyperactive motility, the pattern of motility during capacitation is impaired, very impaired. So with these, we thought that, these ion transporter would be a good target for contraception. It's only expressed in male germ cells of the testes and in sperm. So that will reduce the chances for secondary effects in the body when trying to target it. It's expressed after meiosis, so targeting it should not affect spermatogenesis and should give temporary and reversible infertility. The good thing is if we target it, it cannot be compensated by the somatic form of the sodium pump. And the other advantage is that we could inhibit this alpha-4 either when sperm is in the testes, epididymis, or even after when it's released into the female genital tract. So to do this, we went back and looked at the functional properties of alpha-4. And one inhibitor that is very specific for the sodium potassium ATPase, but it inhibits all isoforms, is wabine. And wabine binds to a specific site, extracellular domain of the sodium pump. So when we study inhibition by wabine of alpha-1 and alpha-4 isoforms expressed in insect cells, and the reason we use this system is because insect cells do not have an endogenous pump, we saw that alpha-4 had a much higher affinity for wabine. And you can see it here in rat sperm. There is the biphasic curve corresponding to the alpha-1 and the very sensitive alpha-4 isoforms. A similar curve can be obtained in humans, and the affinities in humans and rats of alpha-4 is very similar. So this high affinity, wabine affinity property then could be exploited to try to inhibit alpha-4. We also check the effects of wabine and sperm motility. That's sperm and normal sperm non-treated with wabine, but when you treat it with guavain, you can see a decrease in sperm motility with concentrations that already inhibit alpha-4. And if you use more guavain, you do not inhibit much more just because alpha-1 cannot compensate for alpha-4. Same results were obtained in humans. So having these, trying to take advantage of these properties, we started looking at the structure of guavain, which we cannot use because it inhibits all isoforms, and then it would be toxic. So we started with some plant derivatives and and compounds that belong to the same family that guavain belongs to. And guavain's structure consists of 3 main domains: a steroidal backbone, a lactone ring, and a sugar moiety. So we started with compounds that have different sugars, like strophantidine without sugar, simmering with 1 sugar change, and others as digoxin that has several sugar domains. And we found out that the sugar domain does not influence the binding to alpha-4. So then we started working, just changing, and this was all work done by Shamin Shera in Dr. Georg's lab. And we did modifications of carbon-17 and carbon-7 in guavain, and make guavain derivatives, as well as small molecules. So we started with like approximately 30 compounds. Now we have like 60. And it turned out that several of them showed very potent activity in the nano to picomolar range. We have done, homology modeling of the alpha-4 to alpha-1 and look at the docking scores, and they have very good docking with alpha-4. And not as well with the other isoforms, but experimentally, we looked at inhibition curves, and why alpha-4 inhibits with high potency, it doesn't do the same thing with the other isoforms, which are only affected at very high doses of the compound. So we have an isoform selectivity that is very good. The 3 main lead compounds were checked against sperm motility, and as you can see here, they can inhibit sperm motility. This is the control, no treatment, and still there is some movement of the flagellum, but it's highly decreased. Hypermotility was one of the parameters that was affected the most. And these compounds show very good affinity, and even washing the sperm after, there is a little reversibility, at least for the time of the assay. We got similar results in human sperm. These previous results were done in mouse sperm. So these compounds also affect biomarkers that are depending on the function of the sodium-potassium ATPase. And for example, our compounds are able to depolarize the cells, same thing that happens in the knockout mouse. It acidifies the cells through, dissipation of the sodium gradient, and also increases calcium concentration in sperm. That are all parameters that happen in the knockout mouse. We have studied all the properties of the compounds just for their use in vivo, and they have good solubility. They have the permeability of these compounds varies. Some are better than others. So we are trying to improve that too. And in terms of metabolic stability, for example, one of our lead compounds, it's very good, and basically, they are slowly metabolized.
Gustavo?
Yes.
We'll need you to wrap up in a minute or so.
And I will. So cool. We've also did quick tests to show that toxicity was not that high, and then we injected them in vivo. And after injections in vivo, we collected sperm and found that there is a decrease in sperm motility. So these compounds are long and big and difficult to synthesize. So we're working now with a series of smaller derivatives that are showing, also good specificity and activity. So we hope that these compounds could be good for male contraception through the alpha-4 isoform. So I'll finish there, and I thank you very much for listening.
Thank you, Gustavo. Lovely presentation.
Thank you.
Just a reminder to put your questions in the Q&A and not the chat. Does anyone have any questions for Gustavo? Oh, there's a sneaky extra slide there. Very nice.
Well, just in case. Yeah, let me try to go back to the Zoom here. Yeah, I'm trying to look at the chat. Are you going to, oh, no.
Okay, I think you can also read, but I'll read them out loud. So the first one is, to which region of alpha-4 does Wabin bind?
It's to the extracellular side of the alpha subunit of the sodium pump, and the main side is the extracellular domain between transmembrane domains 1 and 2.
Excellent, thank you. A question from Dan Bernard. If these molecules ever become drugs, who would take them, men or women, and will they cross the blood-testis barrier? Well, according to their permeability, the Wabin derivatives, bigger compounds, some have low permeability, but others do, and the small ones also has a much higher permeability. So we are expecting that they could cross the blood-testis barrier. So, but if they don't, they may have an opportunity also to target sperm in the epididymis. So my goal would be to find a pure male contraceptive, but even it could be used on the female, you know, for later, after ejaculation.
Okay, we have other questions coming up, but I think we might have to end there and have the other questions addressed offline subsequently. Sorry about that to those who are asking questions, but we do need to try to keep to time. Thank you very much, Gustavo, for a really, really interesting study.
Thank you very much.
Okay, so I'd like to introduce our next speaker, who is John Amory from the University of Washington, and he's going to talk about inhibition of retinoic acid biosynthesis as a target for male contraception. Thank you very much, John.
Great, thank you so much for having me. I'd like to thank the organizers for inviting me to talk about this interesting work. And this is a bit of a different story. I think most investigators in the area start with a target and try to develop a drug. We actually had a drug, here it is, Win 18446, that was tested as a male contraceptive almost 60 years ago. And it was studied in about 72 men, and it effectively and reversibly suppressed sperm production. There were no pregnancy data from the time. The men tolerated the drug well, and the investigators were very excited that they had a male contraceptive that was reversible. Unfortunately, it caused disulfiram reactions when men drank alcohol. In other words, they were unable to drink alcohol and take the drug. And as some people noted, if you couldn't take the drug, if you couldn't drink, you wouldn't need a male contraceptive. So it really didn't make it any further than that, and it was discontinued. And you can see the reference there. This was a clue to us that maybe this drug was inhibiting an aldehyde dehydrogenase, which is involved in the metabolism of alcohol. So we hypothesized that what was going on with Win is, even though it was interfering with the metabolism of alcohol in the liver, at the same time that it was interfering with the function of an aldehyde dehydrogenase in the testis that was necessary for sperm production. And the aldehyde dehydrogenase in the testis are called ALDH1A1 and 1A2, and these were known to be involved in the biosynthesis of retinoic acid, which was known to be essential for sperm production. And we do know that in particular, one isoform of this enzyme is very highly expressed in the testis, and you can see a southern blot of that here. So given this hypothesis, we developed this animal model in rabbits where we could train them to ejaculate sequentially over time, and we treated them with Win 18446, and you can see that in this box here. And sure enough, the treated animals started with a high sperm concentration, and over about the 12 to 16-week treatment period eventually became completely azoospermic. When you discontinue the treatment with Win 18446 here at the line, you can see the animals take a while to recover, but after about 16 weeks, they get their spermatogenesis back. Concordant with our hypothesis that this was working via the retinoic acid pathway, if you measure the tissue retinoic acid in the testis at baseline and then during treatment, you can see that it's significantly reduced by treatment. And we thought this was good evidence supporting the idea that this drug, WIN18446, was working to inhibit retinoic acid biosynthesis in the testis and therefore spermatogenesis. Here's some testis histology from these rabbits that were treated with WIN. You can see on the left here the normal tubules with normal spermatogenesis. And after 16 weeks of treatment, the tubules look like this. You can see that only the spermatogonia and the Sertoli cells are present and nothing further. And that's very consistent with where we would have expected a drug blocking retinoic acid production to work because retinoic acid is known to play an essential role in spermatogonial differentiation. So in the absence of retinoic acid, you would just expect to see undifferentiated spermatogonia in the tubules, and that's what was seen. We went ahead and worked with Ernst Schönbrunn and his group to try and figure out exactly how WIN18446 was binding to the target. And they did this beautiful crystallization work here. You can see this is a subunit of ALDH1A2, which is a homotetramer. And you can see that the WIN drug fits very, very beautifully in the active site there. In fact, WIN18446 is a mechanism-based inhibitor of the enzyme. And you can see here the drug is actually binding to the catalytic cysteine number 320 there via that disulfide linkage and irreversibly inhibiting the enzyme. So it's a very potent inhibitor of retinoic acid biosynthesis and gave us some clues as to moving forward with drug development. For about the last 8 to 10 years, we've been trying to develop novel specific inhibitors of these enzymes because unfortunately the old scaffold of the WIN, we could not separate out their inhibition of the retinoic acid biosynthetic enzymes, ALDH1A1 and 1A2, and the inhibition of alcohol metabolism at the liver, which is done by an enzyme called ALDH2. So we did the usual medicinal chemistry. We screened a library, and our most promising initial compound that we ended up testing was something called CM121, and you can see the structure here. It's a pyrazole with a methylsulfone group that we thought fit into the active site. The Schönbrunn group did similar crystallography for us, and you can see again here is the subunit, and this time with CM121 in there. You can see that this is a reversible inhibitor, and it has hydrogen bonding rather than covalent bonding to that catalytic cysteine number 320, and here is the stick figure of that. So this was a fairly potent inhibitor of ALDH1A2 with an IC50 of about 100 nanomolar, and it seemed like it had reasonable pharmacokinetic and drug-like characteristics. Unfortunately, this compound did not suppress spermatogenesis in mice. You can see here's our WIN, which serves as the positive control. And you can see that after 5 weeks of treatment, here's the control here, and here's the testis weight. You can see there's about a 70% reduction in the testis weight with WIN treatment, and there's about a 99% reduction in the total spermatid count of the homogenized testis. Unfortunately, here's our WIN group here, and you can see that it really didn't have much of an effect on sperm production. So we had to go back to the drawing board there and think hard about the pharmaceutical characteristics of these inhibitors. Where we've gone to next is we've actually decided, gee, we've got a pretty good scaffold here from CM121 that gives us reasonable isozyme specificity. What if we put this dichloroacetamide group from WIN, what if we put that there, and that results in a compound called CAF75, and I can show you the AC50 curve here. So these are our two target enzymes, ALDH1A1 and 1A2, and you can see that it's pretty good, sub 100 nanomolar in terms of its in vitro IC50 for 1A2 and about 100 nanomolar for the 1A1. And we do think we're going to need to inhibit both isozymes to get a low enough reduction of retinoic acid in the testis to stop spermatogenesis. So where were we with this project? Well, again, we had that prototype drug. What we really needed to do was create drugs that had specificity for the retinoic acid biosynthesis and wouldn't interfere with alcohol metabolism. So we have identified potent compounds without prior patents, but we have been unable to find an in vivo effect that's similar to WIN18446, largely, I think, due to the pharmaceutical characteristics of our current generation. So we're working on just the things Gustavo was talking about, compounds with better permeability, better pharmacokinetics, better tissue penetration, and hopefully we can come up with compounds that will work better than the ones we've tested previously. So if we can do that, we'll continue to model, try and optimize the pharmaceutical characteristics of our inhibitors, go back and do more PK and PD work in mice, and then test the promising compounds. It's handy here. We've got Winn as a control. We know that works. We know it can inhibit retinoic acid production, and we can use it as a positive control in these experiments. If we see good effects in mice, then we can take it back to the rabbits as we did previously, and then hopefully to an IND phase where we can start thinking about testing in humans. And I think my 10 minutes are up. I want to acknowledge all my wonderful coworkers here at the University of Washington who do the experiments. Alex Goldstein is the chemist doing most of the synthesis, and I mentioned Urs Schönbrunn, who did all the crystallization for us. Thank you for your attention.
Right. Thank you very much, John. So I've got one question, or two questions so far. So firstly, when this compound was originally tested, what was the route of administration?
Oral. This was given orally. It was actually discovered during screening. The company, Winthrop Pharmaceuticals, was trying to develop antihelminthic compounds, and this series was actually tested in laboratory animals, found to have reversible suppression of spermatogenesis, and then subsequently tested in men, and it was always given orally.
I'm not sure what that was.
Okay.
John, yeah, I can still hear you fine. I think everything's okay. I'll move on to the next question. Any idea what will happen if the male missed one day of, I assume, consuming this compound?
Yeah, very good question. So since this compound blocks spermatogonial differentiation, it's to me, it's very attractive as a contraceptive because, of course, it takes 70-odd days for a man to make a sperm. So once a man attains azoospermia, it's going to take him a while to restart spermatogenesis. I think as you saw in the graph with the rabbit data, it took about 16 weeks for the rabbits to restart their spermatogenesis. So although we would never say this to the men, it probably affords them actually some degree of missing a few doses once they attain azoospermia, because without retinoic acid, you're not even going to have spermatogonial differentiation, and without that, you're not going to make sperm. So I think there'd be a little bit of a safety blanket, if you will, for men. Other things you could think about were taking a compound that was potent enough and putting it in an implant or giving it as an extended duration shot. Obviously, we need the compound first, but thinking about compliance, I think, is reasonable, especially given potential long-term use of these types of compounds.
Fantastic. Question from Carmen Williams. So there seem to be a large number of ALDH enzymes expressed in humans. Are you concerned about inhibition of these other enzymes by your compounds? And how are you testing for off-target effects of the compounds you're developing?
Yeah, I think that's a very good question. So there's 20-odd members of the aldehyde dehydrogenase family. I mentioned a few of them, ALDH2, which is involved in alcohol metabolism in the liver. There are other compounds throughout the body. So testing for specificity is, of course, very, very important. Fortunately, there's sufficient structural differences between most of the members of this family that we're going to have some innate specificity as we develop compounds. Then, of course, we're going to need to test them, against the whole panel of these enzymes to make sure we're not having off-target effects. And I think that's actually, stepping back, that's a big question for the field in general. I'm sure many of you are familiar with the experience of Gossipol and other non-hormonal contraceptives that looked very promising, but after they were given to a large number of men, side effects were identified. In the case of Gossipol, hypokalemia, and then a failure to recover spermatogenesis. So we're going to need to do a lot of testing of these compounds to make sure that we're not having any untoward effects, both immediately with administration and then long term. So it's going to be a big project to make sure that these compounds are safe to give to people for long periods of time.
Okay, I think maybe this will be the final question. Really interesting one from Joy. So on the flip side, if you use specific ALDH1A2 activators instead of inhibitors, could you boost the number of sperm in men with low sperm count?
I love that question, Joy. So what I do some of my time is treat men with infertility from low sperm counts. So we've actually given them retinoic acid, not an activator as you suggest, because we don't have one of those in our armamentarium, but we do have forms of retinoic acid, both all-trans and 13-cis, that are approved drugs. And if you're interested, maybe you and I can talk another time about the data that we've published from small pilot trials, admittedly, but giving 13-cis retinoic acid to men with oligozoospermia, and in about half of them, you see a significant increase in their sperm production, and you see some pregnancies. So I'm also pursuing retinoic acid therapy, as you suggest, as a potential treatment for men with oligozoospermia.
Really nice, thank you. There are still some remaining questions, but we'll have to move on at the moment. Thanks, John.
I want to answer that one about when in the female. It's been tested, but it's teratogenic. So it's not a good contraceptive for women.
Very good.
Okay, thank you very much, John, for a really interesting presentation. And I'll introduce our next speaker, who is Wei Yan from the David Geffen School of Medicine at UCLA. He's going to talk about SD1 as a reversible non-hormonal male contraceptive agent. Thanks very much, Wei.
All right. Well, thank you, Gerald, for the introduction, and also thank you for the invitation. Today, I'm going to share some of the latest data on the contraceptive development. So I'm not going to talk about how we narrow down and identify this compound, but just tell you this is a long story. And so I'll just tell you that SD1 is a compound purified from a Chinese herb. And so we tested this compound because we, based on the basic research and also our search for different compounds that can cause the sperm deformation. So I just want to give you, I just want to dive into the dry data and just show you what we have got so far. So this is the proof of concept. As you know, like when you develop a contraceptive drugs, and the first step is to, once you identify the compound, you need to do the so-called proof of concept efficacy testing. So this is the efficacy testing we perform on mice. So these are the B6 mice. And so we try the different doses, and every week, and then we set a group of mice and examine the sperm morphology and counts, and also analyze the testis morphology. And so the earliest time we see the deformed sperm with 100% penetrance is week 3. So these are the representative images to show you that this is a vehicle control. This is on the week 4. So it's 27 days and 1 day short. And so these are all normal epididymal sperm. So this is the mice under the SD1 treatment for 27 days. As you can see, 100% of the sperm show this head bent back phenotype. So the head actually bent back 180 degree and with 100% penetrance. All the sperm look like that. Of course, they show a very reduced motility. And so this is 3 months, this is 6 months. So basically, this is the longest time we treat the mice. And so 6 months, just don't forget, that's almost a quarter of the mouse lifespan. But at the same time, so you see this 100% sperm deformation, but if you look at the testes and the epididymis and the gross morphology, we don't see any differences between the treated and the control. If you look at the histology, so this is the control, this is SD1 treated, you don't see really significant, any disruption, any obvious disruption. So they just look like normal, despite the fact that 100% of sperm, epididymal sperm, show this head back, like a deformation. So these are original data that show, as I told you, that we tried the different doses for different time. And then so we call this the minimal effective dose is we identified as 0.8 milligram per kilogram body weight. Of course, you go higher. We also tested the 1.6 and we even tried the 3.2. And there's no problem, but we tend to use we choose to use the minimum effective dose. That's all the data from now on, based upon 0.8 milligram per kilogram body weight. So this is the 3 weeks of data. So you can see actually 100% the sperm got deformed, showing the head back phenotype. And then of course that morphology, motility got significant decrease. Actually, they only show this in situ movement. And so they almost do not have a forward motility. Although the sperm are all affected, if you look at the sperm counts, they the sperm count actually are comparable to the control. So in other words, the sperm output, like the sperm yield, still the same despite the fact all sperm are deformed with reduced motility. And then if you look at testis weight, as I showed you earlier slides, like the morphology, like the testis size did not change, as you can see here. And we also measure the body weight. Usually the body weight change reflects the side effect. We didn't see any significant changes. So this is so basically starting from week 3, if you gave the single oral dose of SD1 at 0.8 milligram per kilogram body weight continuously for 3 weeks, you can achieve this full effect, basically 100% deformation of all the epidermal sperm, while these testis weight and testis morphology had no change. So this is, if you make the milk mice at this time, so they are all infertile. And so if you stop the treatment, and so the male fertility comes back 100%. So this is a figure shows that this is the control. We have 4 mice, 4 male mice, and this is a 1 month treatment. This is a 3 months treatment. And so after 3 weeks of treatment, and then we put fertility proven females into individually caged male mice. So this is like a 10 mice after 1 month of treatment. And then we observe how long it takes to get the pups. So the control, we all know mouse pregnancy takes about 20 days. After 20 days, all 4 controls serve pups. And for the 1 month treatment, 3 months treatment, they all produce pups after about 45 days. We know minus the 20 days pregnancy, this means the recovery takes about 20 to 25 days. So it's sort of the same time frame in terms of achieving the full effects once you stop it and try to get the full recovery of fertility. And then we continue to breed these mice, so this fertility recovered mice, and they show the normal litter size and normal litter interval. So they just behave just like a control. So just to show you a couple of movies. So this is the vehicle control, and so the sperm display the vigorous motility. And then this is SD1 for 21 days. And they only move inside to do the circling. And then there's almost no forward motility. So this type of sperm, they cannot fertilize eggs at all. So this is 27 days. And then we treat it for 3 months. So, and then this is basically the same phenotype as the 21 days. And then this is the recovery. So basically we stop treatment, and then 21 days later, and then you can see that all the sperm actually, the sperm motility, sperm count, morphology, everything comes back. So this is the 6 month treatment. As you can see, all the sperm are deformed, and then they have minimum forward or no forward motility. And after treatment, 21 days, so everything comes back. So it's kind of like a full recovery. And then you do the mating, actually they kind of, as I showed you the data, they're all fertile. So we also measure this is the 4 weeks data of mice, and then we measure the HPT axis by measuring the serum testosterone, intratesticular testosterone, LH and FSH, and we didn't see any significant changes. So here's the thing that people always say that it works on mice, it doesn't mean work on humans, and that's really true. Actually, there were some cases like that. So that's why we went ahead and tested this compound on Cynomolgus monkeys. So these are also called crab-eating macaques. It's very common in East Asia. So we tested on mice and using the similar protocol. And this time we tested, we cannot afford too many doses. We tested 3 doses, and we identified the minimal effective dose is 0.1 milligram per kilogram body weight. So this is about 1/8 of the mouse dosage. And so this is a control, 10 weeks of this is like a daily single oral dose of SD1 on monkeys. So this is the 7 week, actually this is before treatment, this is after treatment 7 weeks, 15 weeks, and then starting from 7 weeks, you can see 100% of sperm actually got deformed. So basically all the data shown here is that, after 5 weeks, you can see significant changes in sperm morphology. And then by week 7, 8 is 100%. So sperm deformation. But the only difference here, the data actually from monkeys are different from the mouse data, is that in mouse, remember I told you there's no changes in sperm count, but in monkeys, the sperm count decrease, as you can see here. And then the motility, of course, like the similar to the mouse data, and basically the sperm showed minimum forward motility. And so for monkeys, or for people who work on monkeys, you know that it's really expensive to do experiments. So we have 4 monkeys. We kept them on SD1 daily, single oral dose for up to 126 weeks. If you calculate it, this basically is 2.4 years, almost like a 2 years and a half. And then basically they do not show any significant side effects, at least we cannot tell. And then in terms of sperm motility, sperm morphology, sperm count, they are like just similar to the 8-week data. So basically you can keep the infertile status of these monkeys for up to 2.5 years. That's the longest time we have tested so far. If you look at testis morphology, actually we did see some changes in testis morphology. This is different from what we saw on mice. And so basically after treatment, you do not see many elongated spermatids in the testes. Other than that, you just don't tell. So that's why the testis weight, actually the testis size decreased like slightly, all right? And then, so we test the reversibility. Basically, we treat the monkeys for 8 weeks and then stop it. And so basically, this data shows that within 4 weeks, all the parameters, semen parameters actually come back. So by 8 weeks, it's completely recovered. And if you do the mating, you test the fertility actually after recovery, they all gain the fertility. And during after 8 weeks of treatment and up to 2 years, we continuously monitor the fertility. Actually, all the monkeys we tested are infertile. So this is the one of the babies born by a male monkey recovered from SD1 treatment after almost 2.5 years. And so this is the things I already mentioned. So this is fertility test. People say you tested too few. I'll tell you that I wish I could have tested 10, but you cannot afford it. Remember, the monkeys that got pregnant only once in a year. They only carry like a singletons. They only one baby birth after 6 months of pregnancy. All right. But anyway, so the preliminary data shows that it's on SD1 you can induce infertility in male monkeys within 7 weeks, and the recovery takes about 5 to 6 weeks. And then once recovered, they become fertile again. So I just want to summarize. So based on our data, we claim that SD1 is a very promising male non-hormonal contraceptive agents. So its bioavailability is really good. You can take it orally. It is effective. As I showed you all these numbers, and it's a completely reversible. And then the safety, I want to mention that we already measured the ED50 in mice. It's like a 0.8. And we also measured LD50. It's a 300 milligram per kilogram body weight. So in other words, if you want to take this pill to commit suicide, you need to take 3,000 pills. So which is unlikely. But anyway, so it's just show that it's quite safe. And we think that the advantages of this compound is that it doesn't cause significant testis shrinkage. Unlike the compound that eliminated all the spermatogenic cell, inevitably you're going to shrink the testis size. And this one doesn't have that effect. So lastly, I want to thank the lab people who performed the work, and most importantly, the funding sources. And I've been fortunate enough to get funding continuously from this company. And recently, I also received funding from MCI to support my pharmacokinetics and toxicology study, and I really appreciate it. Some of you may know or may not know, I recently relocated from Reno to LA. Right now, I'm at the Blanco Institute at Harvard UCLA Medical Center, which is located in Torrance, California. So keep in touch. If you have any questions, I don't have time to talk about other things, and just like offline, we can talk about that. Just drop me some lines so we can talk more if you're interested. So I will stop here. Thank you for your attention.
Great, thank you very much, Wei. We have quite a few questions in just a couple of minutes, so I'll go through them. The first one, did you say that SD1 has a herbal source, and what is that source?
Yes, it's a Chinese herb, and this is one. There are already, if I remember correctly, over 200 compounds purified from this Chinese herb, And this is one of the compound that purify from this herb. So for the time being, I just want to keep it SD1. So I'm trying to publish this paper, and once the paper is out, and then you're going to know the name of the compound, the structure, and everything.
Very good. What do you know about the mechanism of SD1 deformation of the sperm head?
That's a very good question. So we are revising our paper. In the paper, the significant portion of the paper actually reporting the mechanism. So I just want to tell you just briefly, the SD1 actually bond with a very high affinity to a protein called gamma-catenin. There's another name for this protein called junction placoglobin. And so this protein turned out to be interacting with SPAM1. So once you have, in the presence of SD1 bond really tightly to JAP, or this gamma-catenin, and blocking the interaction between SPAM1 and the JAP. So what happens is that the SPAM1 function got completely compromised. That's why the mouse and the monkey phenotype basically phenocopy the SPAM1 knockout mice.
Very interesting. Did you measure whether sperm reached the ampulla in fertility trials, and could the deformed sperm penetrate the zona pellucida?
We didn't test that because they do not have motility. They do not have forward motility. How can they get to the ampulla? There's no way. So so that's why we didn't test it.
And what is the specific target of the compound?
Yeah, so as I said, it's JAP. It's a junction placoglobin, also called gamma-catenin.
Okay. So the mechanism of SD1, I think you really answered that one in another question. I'll move on to another one. How often should it be administered to maintain the effect?
So as I said, oral, every day. You take a pill and continuously for, based on the monkey data, 7 weeks. And then you continue on, and then in order to maintain the 100% deformation of sperm.
Okay, thank you very much, Wei.
Thank you.
Okay, thanks very much, Wei, for another interesting presentation and covering a lot of ground as fast as you could. Now, our next presenter was intended to be Efficacy Basiru from University of Ilorin, Nigeria. Efficacy was with us at the start of our session, but has had an internet connection problem, and I'm just checking, and is currently not online. So we'll jump forward, and Haiqi Chen has been willing to jump in and go next for us. So I'll introduce Haiqi Chen from the Broad Institute of MIT and Harvard, to talk about testicular spatial transcriptome atlas reveals functional dynamics in gene programs during mammalian spermatogenesis. Thanks, Haiqi.
Okay. Can everyone see the slides and hear me okay?
Yes.
Awesome. Thanks. Thanks, Sarah, for the introduction. And also, my name is Haiqi. I'm a postdoc from Broad Institute. Today, I'll be talking about a slightly different topic. Instead of focusing on specific proteins or male contraceptive, I'm going to take a step back and look at spermatogenesis in general through the lens of spatial transcriptomics. This audience does not need introduction into spermatogenesis, but we may all agree that understanding spermatogenesis is really crucial to understanding both the causes for male infertility as well as developing a new male contraceptive. Recently, the single-cell RNA sequencing has really revolutionized our understanding in spermatogenesis in that it systematically profiles the molecular heterogeneity in different testicular cell types, and also helps us to understand begin to understand a little bit about the molecular mechanism that has previously not appreciated. But the thing is single-cell RNA sequencing loses all the spatial context because spermatogenesis exists in this spatially confined unit that we call synovial tubule. So when you perform single-cell isolation, you lose all the spatial information. The spatial context is crucial to understanding spermatogenesis in that all the testicular cell types are organized in a spatially segregated manner. And also for each seminal tubule, you have different stages. In the mouse, we know that there are 12 different stages. So in order to really understand spermatogenesis in its native context, you may not only want to know the cell type information and the transcriptome, you may also want to know the seminal tubule and also the stage information associated with each cells. So today in my talk, I'm gonna introduce you our recent effort to build a spatial atlas for spermatogenesis. And for the interest of time, I'm just gonna focus on one story, how we use this atlas to capture the alterations in spatial cellular context during diabetes-induced testicular injuries. So the technology that we use to build such an atlas is called SlideSeq. And at the core of SlideSeq in this densely packed bead array, and each bead is associated with a unique spatial bead barcode. And when we perform in situ sequencing, we can read out those bead barcodes and associate the XY localization of the bead with the bead barcode. Once we perform in situ sequencing, we can directly put a cross-section of the testis on top of this sequenced bead array. And the mRNA from the tissue will be directly captured by the beads in situ. And then you perform a sequencing library preparation that is quite similar to that of a single-cell RNA sequencing library preparation. And at the end of the day, what you get are the two components. The first component is a gene expression matrix that is quite similar to that in the single-cell RNA-seq data, in which you have the gene expression profile for each SlideSeq bead. And the second component is a bead localization barcode that file that we obtained earlier from the UCLA sequencing. And those two matrices are connected through their shared spatial bead barcode. So once we have those two files, we can begin to build up the spatial atlas. As I mentioned earlier, to do so, we need three types of information. For each SlideSeq bead, we need to know its cell type. We also need to know which seminal tubule it belongs to. And furthermore, for each tubule, we need to know which stage it's at. So to start, we use a customized algorithm called NMFREG to automatically assign the spatial and cell type information to each SlideSeq bead. Here I'm showing you a totally digitally reconstructed cross-section of the mouse testes. You can already see the tubule structures quite clearly across this digitally assembled cross-section. When you look at individual cell types and their spatial localization, our assignment is quite accurate in that, for example, the spermatids are localized in the center of each tubules, whereas the spermogonia are localized at the periphery of each tubule. If you look at the somatic cell types, for example, the Leydig cells, they tend to cluster together in this interstitial space in between tubules. So once we have the cell type information, the next thing we want to know is what tubule is the SlideSeq bead belongs to. In this case, we realize that by treating each SlideSeq bead as a pixel, we can effectively turn the genomic sequencing data from the SlideSeq experiment to image data. The reason we wanted to do that is because we realize that in doing so, we can convert the tubule assignment problem into an image segmentation problem to segment out each seminal tubule that we saw in this data. And just by applying a standardized image analysis pipeline, we can group out the beads and assign them to each individual tubules. So once we have the tubule information, the last information we need is a stage. So in this case, we first group the gene expression for all the beads that belong to the same tubule, and then only look at the genes that are most highly variable in the gene expression profiles. And we use unsupervised clustering to cluster them in a gene expression space. Already you can see that tubules form clusters. That's because the tubules belonging to the similar stages should have similar transcriptional profiles. And then we can plot genes whose gene expression patterns has been previously shown to be stage specific to assign stage information to those tubule clusters. So in total, we assigned 4 major stage clusters to those tubules, and then we can map the stage information back to the original spatial data. So to summarize, we have built up a spatial atlas for spermatogenesis in that we assign each sexy bead with the cell type information, the tubule information, and also for each tubule, we assign the stage information. So now that we have this atlas, we can begin to understand spermatogenesis from a different angle. In this case, I'm trying to use this atlas to systematically profile this tissue structures in seminiferous tubules under perturbations. Because male infertility and subfertility is one of the major complications of diabetes. So we're interested in to look at diabetes-induced testicular injuries. So at the cellular level, you can already see that the tubule structures in the diabetic samples are not as clear as those in the wild type. And at the gene level, we also observe a disruption in the spatial gene expression pattern for some of the genes in the diabetic samples compared to that of the wild type. If we zoom in into individual seminiferous tubule, what we found was that the disruption in the genes spatial gene expression pattern was actually largely driven, if not entirely, by a disruption in the spatial arrangement of the different cell types in the seminiferous tubule. So we want to systematically characterize these changes in spatial arrangement of cell types. To do so, we came up with quite a simple matrix we call purity score. So basically what this score measures is the extent of the spatial mixing between elongating spermatids with other cell types. So for example, in a wild type tubule, the elongated spermatids or the blue beads tend to cluster together near the center of the tubule, therefore having a high purity score. Whereas in a diabetic tubule, you'll be able to see that the blue beads tend to spatially mix more with other cell types, therefore having a relatively lower purity score. And then we can calculate the purity score for almost 100 tubules from both the wild type and diabetic samples. We can see that there's a significant decrease in the purity score in the diabetic seminiferous tubules compared to that in the wild type, suggesting that the disruption in the spatial patterns of the different cell type structures are disrupted, and this phenotype is quite widespread in the diabetic samples. So to sum up, today I've shared with you our recent effort to build a spatial atlas for mammalian spermatogenesis. And we showed that we can use this atlas to systematically profile the tissue structures in deep cell types during perturbations. In fact, we have used this atlas to look at other aspects of spermatogenesis. For example, we can systematically identify genes with non-random distribution at the level of seminiferous tubules. We can also begin to uncover the stage-dependent gene expression patterns in different cell types, because as I showed earlier, we already assigned the stage information to each tubule. And finally, we can use the spatial data to infer temporal dynamics of the gene expression and signaling pathways due to the cyclic nature of the spermatogenesis. I think my time is up, so I'm happy to share with you guys this information in another occasion or offline. And finally, I'd like to thank all the members from the Chen Lab at the Broad Institute who have contributed to this project. And I'd like to thank my funding source. The Male Contraceptive Initiative has previously provided me with a travel grant, and also I'm supported by the Leila Foundation Postdoctoral Fellowship. Thank you for listening. I'm happy to take any questions you may have.
Fantastic presentation. Thank you very much. You communicated so much in such a short amount of time. Very impressive. So we've just got time for a couple of quick questions. The first one from Nathan Law. So what is the resolution of the bead array for slide-seq? How much area is actually captured by a single bead?
Yeah, that's a very good question. So the bead array is 3 square millimeters. And for each bead, it's about 10 micron. So that's the size, basically the size of an average cell. One of the challenges that we have is that because I guess the advantage is that because the bead is densely packed, the resolution is about 1 to 3 cells per bead. And I guess that also poses a challenge in that it's possible that one bead can capture transcriptome from neighboring cells, like 2 or 3 cells together. So this makes it challenging to computationally accurately assign transcriptome information and cell type information to each bead. So this is where the NMF comes forward, so that we can decompose the data and accurately assign cell type. And in the meantime, the lab is actively working on refining and coming up with new ways to assign cell types more accurately. And I'm happy to share additional information with you guys offline.
Lovely, thank you. Question from Carmen Williams. So the structural integrity of the OB/OB testis appeared disrupted. Did you need to adapt your algorithm for assigning cells to different tubules so that it would work for the diabetic mouse? And if so, how does that change your conclusions?
Sorry, can you actually repeat that question? I'm a little bit...
Yeah, of course. Also, if you want to go to the Q&A box just down the bottom, you can also read along while I read that. Maybe that's easier for you too.
I think I'm
That's okay, I'll just read it.
Sorry about that.
No problem. The structural integrity of the OB/OB testis appeared disrupted. So did you need to adapt the algorithm for assigning cells to different tubules so that it would work for your diabetic mouse? And if so, does that change your conclusions?
I see. Yeah, that's a really good question. So the question, I guess the answer is it definitely poses a challenge to assign tubules. If the overall structure is really disrupted. But in our case, we found that it's still doable to assign tubules using our current computational pipeline. Because I can talk a little bit about how we assign tubules. So basically, we assign a pseudotime value for each cyclic bead based on the development trajectory. And we can segment out the tubule based on those so-called pseudotime data. And as long as the development trajectory of the germ cells are not disrupted entirely, we can still relatively accurately assign tubule information. So if you can imagine there's a perturbation that totally disrupts the entire development trajectory of the germ cells, then I guess the pipeline would not work very well. Mm-hmm. Hope that answers your questions.
That makes sense. So, question from Dan Bernard. Do your data confirm or challenge the current dogma that there are 12 discrete stages of spermatogenesis in mice?
Well, I mean, we definitely not an expert on that part. And also, I guess the reason we can assign stage in the first place is because the previous amazing work done by a lot of people to show that genes have stage-specific patterns. Therefore, as you can see that the resolution is not high enough for us to assign all 12 different stages, because right now we are only able to assign stage clusters. That's because people has not been able to find a gene that only expressed in just one stage. So this raises an interesting question of, in biology, do we really need that many stages? Because because of the continuousness of the spermatogenesis, you may be able to group adjacent tubules altogether that belong to the similar stages, which is enough for us to assign biological meaningful information. But again, I cannot see at all that our data is challenging or not the current assignment of stages.
Maybe in the future. Okay, I think that's all we've got time for. Thank you very much.
Thank you so much.
Yeah, thanks very much, Haiqi, for bringing that really interesting research to this meeting. That was fascinating. So we are still having trouble getting Efficacy to connect online, so we'll keep trying and add him at the end if possible, and if not, then we'll try to get a recorded presentation and make that uploaded and available online for people to view anyhow. So we'll move on to our next speaker, who is Max Lyon from Washington University School of Medicine. And Max is going to talk about the sperm-specific potassium channel SLO3 is a target for contraceptive development.
All right. Okay, thank you. So to start off, I'm just going to give a little bit of background on sperm membrane potential. So one of the processes that occurs commonly in mammalian sperm as they capacitate is their sperm membrane potential hyperpolarizes. So in mice, what this means is their membrane starts at a resting potential around -45 millivolts And then during capacitation, it becomes increasingly negative, ending around minus 65 millivolts. And we know that the channel responsible for this change is SLO3. SLO3 is a potassium channel, which is expressed exclusively in sperm of mammalian species and potentially garfish and some others. And in sperm, it is responsible for a potassium current, which is activated by intracellular pH. And if you knock the SLO3 channel out, you lose that pH-activated current. When the SLO3 channel activates during capacitation, it causes a potassium efflux, and the loss of positive charges inside the cell causes them to become more negative, which is what drives this hyperpolarization. If you knock out the SLO3 channel, you lose that potassium efflux, and this means that the sperm no longer hyperpolarize. Downstream of this, the sperm lose the ability to get a calcium influx, which is important for several downstream processes. And as a result of that, they have impaired motility. They also fail to undergo an induced agrosome reaction, and they ultimately are unable to fertilize. And what this means is that the SLO3 function of hyperpolarizing the sperm is necessary for fertility. Now, in human sperm, the SLO3 channel is also present. It seems to have a similar function, and its modulation is slightly different. It's more activated by intracellular calcium than intracellular pH, although it is still pH sensitive. And while there are no known human loss of function mutants, there are known genetic variants which have increased pH and calcium sensitivity, indicating that it is still physiologically relevant. And all this together indicates to us that it is a good target for contraceptive development. However, no selective inhibitors for human SLO3 have yet been described. Thankfully, SLO3 is able to be stably expressed in heterologous systems, and this means it's able to be used in high throughput screens. So our partners at the Denton Lab and Vanderbilt University developed such a screen using a cell line stably expressing SLO3 and the auxiliary subunit LLRC52, which is known to also play a role in fertility. And the way this screen works is they have the cells expressing SLO3 loaded with a dye called thallose, which binds to thallium, which is a monovalent cation permeable through SLO3 channels. As the thallium binds the dye and fluoresces, they can add small molecules, and if they inhibit or activate the channel, you'll see a change in the fluorescence based on the SLO3 activity. They have already screened over 50,000 compounds from a library at Vanderbilt, and then gone through several different selectivity and screening steps, and then given us the resulting compounds from this to validate using patch clamp electrophysiology. Just a brief overview of patch clamp. The way it works is you take your cells expressing your channel of interest. You attach a small glass pipette, break the section of the pipe or the membrane connected to that pipette, connecting it to the cell, and then using an electrode in that pipette, you expose the cells to a series of voltages, and then as you drive the current through the channel, you're able to record that and then see how that channel is modulated based on various factors. So we expose these cells to a concentration gradient of our inhibitors, and I'm only going to focus on one of them, which we've designated 110, and saw a decrease in the current. We then use this to generate a dose response curve and calculate the IC50 at about 767 nanomolar. Now, the compounds that are currently used to examine SLO3 are typically used in the 100 micromolar range to see this type of inhibition, and they are non-selective, which means that this inhibitor is far more potent than what we have seen previously. We also wanted to make sure that the inhibitor 110 selectively targeted the SLO3 channel, and the first target that we looked at was SLO1. SLO1 is very closely related to SLO3. It has been proposed to be present in human sperm, and it's ubiquitous throughout the body. So if we are trying to develop this compound as a contraceptive, it's very important that we don't target SLO1. And we do see that there is no effect on SLO1 at low concentrations. We also wanted to make sure that it wasn't interfering with any subunits which are shared between SLO1 and SLO3. And two of these that we tested, LLRC52 and beta-4, are present in testes and affect both SLO1 and SLO3. And once again, with the SLO1 co-expressed with those subunits, we did not see any effect at low concentrations. The next step was looking at the effect on sperm membrane potential. And in a paper that we published this year, we saw that the fertilization rate of human sperm was correlated with membrane potential. So if you can prevent this hyperpolarization or cause the sperm to depolarize, you could theoretically reduce fertilization. And we used two different methods to look at this. Both used the dye DIS35, which is a positively charged dye that distributes based on membrane potential and can be used to calculate the membrane potential of human sperm. And in the first experiment, we looked at what happens when you incubate the sperm at a high pH, which we know causes a depolarization of human sperm. When we incubated sperm at pH 8 with inhibitor 110, we saw that this depolarization, or that this hyperpolarization did not occur. Next, we used flow cytometry to examine sperm that were incubated in capacitating conditions. And when we added inhibitor 110, we saw a decrease in fluorescence, indicating the sperm were depolarized. Finally, we looked at the two hallmarks of capacitation, which were hyperactivated motility and the agonism reaction. And when we incubated sperm in capacitating conditions and then added inhibitor 110, we saw a significant decrease in the hyperactivated motility. And when we incubated the sperm with the inhibitors in capacitating conditions overnight, we saw a reduction in the induced agonism reaction. With our sample size, this wasn't significant, but it was trending lower strongly. So in conclusion, we identified the first novel and potent selective inhibitors for SLO3. And we found that this inhibitor inhibited hyperpolarization, hyperactivation, and the agonism reaction. And we're currently in the process of developing second generation inhibitors with increased potency and selectivity for SLO3. All right, I would like to thank my lab and our collaborators and then open it up to questions.
Great, thank you very much, Max. We've got one question so far from Barcadella. So does patch clamping work on mature sperm, or is it the cytoplasmic droplet containing sperm?
Yes, so in human sperm, the ejaculated sperm actually do contain the cytoplasmic droplet right at the neck. In mouse sperm, the cytoplasmic droplet is at the end of the midpiece and typically dissipates as the sperm mature. So in patch clamp experiments on sperm, you typically patch the cytoplasmic droplet, which means that in mice, you usually try to patch an earlier maturation stage than you need to in human sperm.
Okay, that's really interesting. How are you planning to increase the potency in your next generation?
Yes, so good question. Our collaborators at Vanderbilt, they have a medical chemist on their part of the project, and they've been developing structural variants of the channel, specifically designed to reduce the off-target effects based on the structures of the off-targets.
Okay, fantastic. Do we have any further questions for Max? From Naomi, just out of interest, did you also assess intracellular calcium levels?
So we have some preliminary data on that, and we did see a slight reduction in intracellular calcium. That is something that we are looking forward to pursuing more, though.
Very good. Max, I just picked up in the first couple of slides, you said that this potassium channel is sperm specific. I'm really surprised that there aren't very similar channels in somatic cells.
Yes, so SLO1 is the most closely related channel to it, and I believe evolutionarily it's thought that SLO3 came from a gene duplication of SLO1, and SLO1 is expressed ubiquitously, and it's very important in many other systems. And there's a lot of bleed over with compounds that affect either channel, which is part of the reason why it's difficult to find things that target specifically SLO3. And that was why we spent so much focus on looking at the effects on SLO1, because we wanted to make sure if we're developing contraceptive, you're not getting damage to the brain or the heart or various other tissues.
Oh, very interesting. One more question from Bart. How does SLO3 relate to cat spur? Is it downstream? Yes, so in mouse sperm, cat spur is upstream of SLO3. So if you knock out SLO3, you lose cat spur activation, and that's thought to be one of the main reasons why you see the defects in the agonism activation or agonism reaction and motility. And if you bypass SLO3 by activating cat spur directly, or like you had valinomycin, which also hyperpolarizes the cell, you resume cat spur activity and you get those downstream processes still occurring. In human sperm, it's less clear, partially because of the calcium activation of SLO3. But based on the results that we saw where we inhibited SLO3 and saw downstream effects, we think it might be a similar role in activating cat spur. Very interesting. Do we have time for one more? I think we might.
Cool.
How effective is 110 in preventing pregnancy in animal models?
So we have not tested that yet. We have some preliminary data in mice using both patch clamp in the channel expressing oocytes, and then also some preliminary IVF data. It's not clear what the effect is quite yet, and part of the reason for that is the inhibitor seems to have a lower effect on the sperm channel. Human and mouse SLO3 are very divergent channels. They're about 50% sequence similarity, or I think it's, sorry, 60% approximately. And they are both regulated differently physiologically. So we're pursuing the animal experiments. We have a grant to test that further with actual fertilization assays, but we don't know quite yet what the effect is in animal models.
Great, thank you very much, Max.
Okay, thanks very much, Max, for a really interesting presentation. So we still haven't been able to re-establish our connection with our final speaker, Basiru. Now, Efficacy was online at the start of the session, so something went wrong along the way, and unfortunately, under the circumstances, we'll have to end the session without that presentation. As I said, we will try to establish a recorded presentation to make that available, along with all the other recordings of these presentations on the SSR website. So I'd like to thank all of the speakers for some really tremendous presentations. I want to remind everybody that there is another session in this conference that will occur at the same time next week, a little more focused on female-targeted contraceptives, but we again have 7 really, really interesting looking presentations ready to happen next week. There's also poster presentations to be viewed online. And I want to remind everyone to register for the virtual happy hour that will start at the end of the conference session next week. And that's where we will announce awards for both the abstract submitted oral presentations and the poster presentations. Thank you all for attending. I want to thank the SSR's business office virtual for all of the help that they do in making all of this go so smoothly and making it possible. And immediately after this webinar, you'll see a survey appear that we would ask you please to answer and give us your feedback on the conference so far. Thank you very much. Thanks to all the speakers, and there's the survey. I hope you can see it.
Thanks.
All right, thank you. See you next week.
Okay, I think we are